Hello, everybody. It's Mr. Cadell. We're back. We're in the middle of chapter 9, and we are in the thick of things right now. The goal, remember, is to take carbohydrate, like this representative glucose molecule, break it down into nothing, exhale the carbons and oxygens as CO2 out of this thing, and harness all of the energy that was once stored in these bonds. We're going to transfer it to ATP momentarily until that then is broken in order to power us and all other organisms through their day. Don't lose sight of the fact that this was food. We're going to combine it with oxygen eventually in the cellular respiration formula and equation. But we need to harness all this energy. So that's what we're doing. We are in the middle of chapter 9, as I said, and we've gone over glycolysis. Here's the transition reaction. There's a great bit of information here, and I want you to read every word of it. I've told you, if you look at this here, that I want you to take your time and really focus on these figures. Read everything here. I think it's all good and it's all relevant. I just liked my more simpler pictures better. Once you get over here to 9.4, I say here just skim the writing in 9.4. Make sure that you spend a ton of time on these figures. And, I would like you to read it, of course. I just think it's at a little bit, fractionally too high of a level for this 100-level college class. This is fantastic stuff right here. Here's this nice big summary. I would spend a ton of time on figure 9-15. I think the problem is that as you get down here, it starts to talk to you about how many ATPs are made per NAD and FAD. All this stuff is really good, and I want you to read it. I'm just, unlike every other word in this textbook, which I'm going to hold you to on the tests, I just think that this is a bit too high. So if you're skimming this and reading this and it's just not registering as you get to the end of 9.5, that's fine. Or 9.4. It is what it is. I will get you the information today of what you need. As far as 9.5 and 9.6 go, we'll do that the next time we get together. So let's start with the song. In the matrix of the mitochondria of all the cells is the site. I'm just kidding. We've already done that song. So we need to take a look at this, and I want you to remember how we are going to deliver this energy from these bonds to the electron transport chain. Go ahead and tell your neighbor, please. Well, the answer, of course, are these NAD and FAD molecules. They're going to take these hydrogens off, and they're the only thing that matters when it comes to energy here. You can see all these NADs and FADs and where we've seen them before in glycolysis and the citric acid cycle. You can see them. There's a ton of ATP made on the far right side there in oxidative phosphorylation. We'll go over what that means today. Here is the big picture. I'm trying to put this all together. We have glycolysis, then the Krebs cycle, and then at the bottom is the electron transport chain. It all is kind of formulaic, and they all depend on one another. You remember that in glycolysis, the NADs grabbed a couple of those hydrogens, and then in the Krebs cycle, they grab the rest of the hydrogens that they're in the link reaction. Let's do this. Before you go any further, press pause and try to do this from memory without looking at the picture on the screen, and I'll try to do it also. I may or may, I think I have this right. What I've done here is I've tried to do it per glucose molecule because I told you in the last video that you might see that on the exam. I get it if you just talk per pyruvate or per acetyl-CoA once it comes out of the link reaction. We can do that math, but we can also do the math when we say, well, we once had a big glucose molecule. What are the total products that we're getting here as a result of 2 turns of this Krebs cycle, which occur with 1 glucose molecule being broken down? And so I've just doubled all these numbers essentially from the last time. So I just want you to be aware of that. You might see it on the exam. Let me know if you have any questions about this. Oh, one other thing I put in here, which I didn't have last time, is oxaloacetate. It is the 4-carbon intermediate that exists at the end of every citric acid cycle. Sorry, I guess at the beginning of every citric acid cycle, because it binds to the acetyl-CoA to make citric acid. But then at the end, it's also present. That's why it's not only a reactant, but it's also a product. Let me know if you have any questions, please. Let's go here. I want to make sure that you have this piece of paper. We're going to come back to this. We're going to dive into the books pictures real fast, and then I will come back to this. But make sure that you have it. That is the level at which I want you to know. Here is the title of concept 9.4. It says, during oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. We'll get to what oxidative phosphorylation means in a minute, as well as this term chemiosmosis. We'll get to that. But we know that we're going to make ATP at the end here, and we know that electrons are going to be transported down the electron transport chain. In this picture, that is from left to right, going down all those blue things. You can imagine a little electron kind of, I guess, squiggling its way down these things. Remember, the individual that's going to deliver these electrons to the electron transport chain are the FAD and the NAD. So here's an example of NAD. Remember, it accepted electrons in glycolysis, the link reaction, and the citric acid cycle when the hydrogens were being torn off the glucose that was being broken down. So it accepts electrons because they're a part. Remember, electrons are a part of a hydrogen atom. But here's what's new. They also donate. So they are at once reduced when they get the electron, and then when they get rid of the electron, they will be oxidized. So they are going to, these NADs are going to actually get rid of an electron as well, and you're going to see that they're going to give it to all these proteins that exist right here in this inner membrane of the mitochondria. So they are a donor as well. Well, if you recall, every NADH is able to give 2 electrons and 1 proton to this chain. Now, the chain isn't interested in the proton, so I'm going to tell you what the proton does is it diffuses through the membrane when this happens, and then they start to build up on one side of the membrane. Sorry, that's not true. They don't diffuse. What happens is what you can see here from left to right is as the electron kind of goes down from left to right in this energetic situation down the electron transport chain, all these proteins from left to right, that energy is used to suck electrons against their concentration gradient, which means they're going to be kind of put in an area that they don't like. They're eventually going to flow through an opening, and we're going to try to harness that energy. And I'll show you exactly what that looks like with a different picture. This is important. As you see on the y-axis here, that it is free energy. So as the electron goes from left to right down these proteins in the electron transport chain, you can see how they're losing energy. I guess on the x-axis would be time. They're losing energy over time as they get kind of traded from these proteins. So let's take one protein. So I see CYTB, so that's cytochrome B. So what's going to happen is there's a protein in this chain of proteins. It's going to be reduced because it grabs an electron as it's coming down from left to right in this chain. And then it's going to be oxidized as it releases the electron to the next protein in the chain. And that happens, we'll see a movie in a minute. You'll literally see them grabbing onto an electron. They hold it momentarily, and then they get rid of it, and then they turn back and they grab the next electron. So it's a chain, or it's a flow of electrons from left to right here. Well, the whole goal of this is to not have these inefficient combustion reactions occur. So there's a lot of potential energy here, and in spontaneous combustion of a human or combustion in an engine, it's super inefficient. Like 25% of this energy is actually kind of harnessed in order to do work. But what happens in this situation, I've read before that it's 71% efficient at taking the energy from here and putting it into the third bond between phosphate groups on an ATP. So adding a phosphate group to ADP to make ATP essentially. So it's very efficient compared to inefficient combustion reactions. We've evolved to have this. Would I love to see it be even more efficient? Yes. Where is that extra energy going? It's lost as heat. That's why you're hot during your day, and you get hotter when you run around the track and you break down more food in order to power your legs to run you around the track. So it's nice and controlled. And I'm going to read this. It says, in the cristae of the mitochondria, the chain's components are proteins which are alternately reduced and oxidized as they accept and donate electrons. Remember, the cristae is these kind of folds on this inner membrane. There's two membranes on a mitochondria. There's the inner membrane and the outer membrane. And this is the inner membrane space, and this is the matrix of the mitochondria where the citric acid cycle and link reaction occurred. Why don't we do this right now? So go ahead and draw with me. I would say get out a big blank piece of paper. So we have glycolysis out here. Remember that we have a glucose, and that's turned into 2-pyruvate. Well, if oxygen is present and everything looks good, there's a mitochondria here. These pyruvates come into the mitochondria, and then they go through, I'll write here, the link reaction. And at the end of the link reaction, you have acetyl-CoA. Remember then that the acetyl-CoA will bind with something called oxaloacetate, and it will turn into the 6-carbon compound citric acid. This whole thing is a citric acid cycle. And then the whole goal here is to get rid of the carbons and the oxygens. These things diffuse out of the mitochondria. And remember, different color. We've got these NADs and FADs, which get reduced, meaning that they get the electrons from hydrogen atoms they tear off of your food. Okay, fine. So now what happens? Well, what we're seeing in this picture a little bit, and we'll see it in a little more detail in a second, is that the NAD and FADs are going to deliver their hydrogens to this inner membrane here. Essentially, what they're going to do, because you know that this hydrogen is made of an electron and a proton, they're going to, I'm just going to draw a bunch of squares on this, representing all these little proteins that exist in this membrane. The electrons from the hydrogen atoms, which are dumped off by these guys, will go through these proteins. The energy that's harnessed by these guys doing this enables the proton, so these are the two components of a hydrogen atom, enables a proton to be sucked through here, it's supposed to be an arrow, against its concentration gradient. So the idea is that we're then going to build up a bunch of these guys in here. I'm just drawing a bunch of H pluses. So these guys are going to be pissed off because they're like, well, we have the same charge, and like charges repel, and we're next to all of our friends. They're exactly the same as us, and we want to get away from them as through diffusion. And so eventually, an enzyme called ATP synthase or synthetase will open, and it will be basically a chute. And I'll show you what that looks like, kind of the latest imaging that I have in this PowerPoint. It opens, and these guys then are like, sweet, that's my chance. I'm going to shoot through here. And when they do, that energy is used to harness the addition of an inorganic phosphate to a diphosphate, adenosine diphosphate, to make ATP, or adenosine triphosphate. It's this energy of this thing, it actually spins here. This is called the proton motive force. Basically, it's the protons moving this ATP synthase thing around, and it was this force set up by this gradient caused by these electrons kind of flowing through these proteins in the membrane. And then what happens is that the H plus will come back here. Here's his electron friend. You know what an E negative and an H plus, or electron and a proton equal? They equal a hydrogen atom. And so I told you this the other day. Let me just do this. So I'm just going to write an H next to this. So now this is a hydrogen atom. So the idea is that you have to get rid of these things. If these things build up right here, you don't really have this proton motive force to spin this thing to phosphorylate ADP. So the way that you get rid of this is by rebinding it with these electron friends, but also by breathing. And so when you inhale the atmosphere, you mostly get nitrogen, but some of it is oxygen that gets into the lungs, into the bloodstream, diffuses out through the bloodstream to all the cells, diffuses into the mitochondria. And the point is that these will come here, and these individual oxygen atoms of the oxygen molecule will bind with hydrogen atoms to make water. And so people will say, well, what's the final electron acceptor here? Well, the final electron acceptor is oxygen, the oxygen atom, in order to make water. And so that's how all of this works. Like, I know you probably can't see it, but I have a formula up here for cellular respiration. You can see that all the reactants and the products in that formula are represented here. It's pretty amazing. Take a moment. Let's pause for a moment and talk yourself through this, or talk a neighbor through this, please. As your electrons drop in free energy as they go down this chain and are finally passed to oxygen once they rebind with their proton friends to make hydrogen atoms, and then water is made. Press pause, look in the book, rewind, whatever you have to do. Come ask me for help. Look at the pictures in the book. Watch a movie on it, which I'll show you where a couple movies are. You can't skim over this stuff. And that's really what all the languages in 9.4 is talking about, this. I'm just trying to break it down for you. Okay, well, let's simplify. So let's read at the bottom where it says figure 7-5. Hydrogen ions released from NADH and FADH molecules pass through the electron transport chain within the inner mitochondrial membrane, generating a high concentration of H plus on the outside of the membrane. As the H plus passes back through the ATP synthetase molecules within the membrane, the energy from the flow is used to produce ATP. I would have someone else read this in class, but let's keep going. I'm in the top left box. It says electron transport. NADH is oxidized by passing its electrons as part of hydrogen atoms to the electron transport chain. The electrons make 3 round trips, or only 2 trips if FADH2 is the source of electrons, through the membrane, accompanied by hydrogen ions on their outward journeys only. This builds up a high concentration of hydrogen ions outside the membrane. Inside the membrane, oxygen takes the spent electrons plus some hydrogen ions to form water molecules. Chemiosmotic synthesis of ATP. Hydrogen ions move down the concentration gradient inward through channels associated with ATP synthetase enzymes. The energy from this flow of ions is used to make ATP from ADP. Basically, this picture is just showing you exactly what we did here. So press pause, take a moment, make sure you understand it. If you don't understand this, ask me for help. Spend some time in the book, call a friend, whatever you need to do. But you can't push on if you don't understand this. This is a major part of this course and a major part of the exam that's coming up. Well, this is the picture from the textbook, and I'm going to tell you that it's flipped. And so all of this stuff that is occurring in the mitochondrial matrix on the last picture on the screen as well as on the board is on the top, all the making of the ATP and the oxygen coming in. But all that's happening on the bottom of this picture, and that's okay. You could just imagine that they're representing a electron transport chain that's just up here. No big deal. I will tell you that's why the the inner membrane is so folded on mitochondria, which they call the cristae, is to increase the surface area so that more of these electron transport chains can take place. So we just represented one on the bottom, but if you were to, I guess, float upside down, it would be on the top, or it's on the top here. So just understand what we're looking at here is the inner membrane space is on the top of this picture, and then the matrix of the middle of the mitochondria is on the bottom of this picture. I keep pointing at the screen, but it's on your screen right now. Well, we've gone over the proton motive force. I'm going to tell you this whole thing is the oxidative phosphorylation. So think about what's happening here. We're using oxygen, and eventually something's going to be phosphorylated. So I'm okay with that. We'll get to chemiosmosis in just a second. So you can see here that in the matrix of the mitochondria, which is on the bottom of this picture, that NADs and FADs are unloading their electrons, which are going through this membrane. The protons are getting pumped against their concentration gradient into this big pool in this inner membrane space up here at the darker orange in your picture. Note how the H plus ions go to that one side. The energy stored in an H plus gradient across a membrane couples the redox reactions of the electron transport chain to ATP synthesis. Redox just means reduction, oxidation of all these little proteins that exist in the membrane. I'm going to tell you, you don't need to memorize the names of the proteins. You can always look those up. You're going to see them in the textbook and in the CliffsNotes book and stuff, but don't memorize them. Sure. See that a increased number of hydrogen ions are created, a gradient is created on the top side of the membrane, and then all this high concentration of these protons come rushing down on the right side there. That energy is harnessed to make ATP.
In the mitochondrion, the energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the inner membrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen. The terminal electron acceptor and water is formed as the product. The transfer of protons to the inner membrane space generates a proton motive force across the inner membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
First thing you need to know is that you don't need to memorize all of those names of those proteins. I've said that before. Number two, we're only going to do this twice. Luckily, the textbook and the course says that we only have to memorize, or we only have to deal with this twice. And the other one is photosynthesis, which is in the next chapter, and it's actually much easier, in my opinion, than this, because we reuse some of the concepts.
During glycolysis and the tricarboxylic acid cycle, oxidation of organic molecules results in production of reduced coenzymes such as NADH. These coenzymes transfer hydrogens to the electron transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton and an electron. The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen. Electrons enter the electron transport chain when NADH transfers its protons plus electrons to a membrane-embedded carrier protein. The electrons are sequentially carried along the electron transport chain, while the protons are shuttled to the outside of the membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force. During aerobic respiration, the last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed.
If you need more, Mr. Anderson has some really good videos here. We're going to do a couple of housekeeping items. Last thing is the proton motive force, which is what's used to turn the ATP synthetase enzyme and then phosphorylate ADP with an inorganic phosphate to make ATP. I'm going to show you what this looks like with this kind of digital imaging here. Just understand it's not a chute, it's a turbine. Essentially, it's going to turn. You can see a lot of rotational arrows here.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in the proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft. Like any enzyme, ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane. To show the rotation of the central shaft, a short fluorescent actin filament was experimentally attached to it. Single filaments attached to single F1 ATPases can be visualized in the microscope. When ATP is added, the filament starts spinning, directly demonstrating the mechanical properties of this remarkable molecular machine.
This was first highlighted in Stanford, and they saw these little lollipop heads. They were trying to figure out what this looked like. I want you to be amazed when you, please, 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 I've said this before, let this wash over you with amazement. How complicated this is, how all those amino acids that are kind of alpha helixing and beta pleated sheathing inside that ATP synthetase head there, how they're all perfectly arranged, how phosphate is added to an ADP to make ATP. Be amazed by this, and then just understand that it's like one out of thousands of different types of proteins that we have in our body. I just think it's amazing. Here's my final video. Just it's a picture of this essentially spinning around. I don't know how we've evolved to have all these wonderful, complicated and efficient things. I mean, I guess we have evolved to have them. I just think it's wonderful. I just really do.
Yeah, let's see the snap. High schooler was confused by oxidation of food. Seemed too complex, like how to have me confused. But it's a simple concept, so now we move forward, learning how I'm burning glucose as a food source. I want to live, dressing my cells, take energy from the bonds and the sugar, put it in the ATP. Takes 10 enzymes, takes 10 steps, line up to my root base, yeah, still got energy left. So mitochondria can take them to burn, the downward cyclical is correct on terms, so the energy you get from these damn blueberries is very the high energy electrons to carry. 8 to 11, I call it Cisco chop. It comes down to go ahead, study me. I'll make ATP, and I'm gonna be on your test, so you should get to know me. And if you got enough oxygen, then stop. There's no cycle, I'm gonna be into your heart stop. Go ahead, study me. I help make ATP, I'll make it in ATA, so you should get to know me. Hydro. In the membrane of the mitochondria, using NADH to press pump protons, mixed with electrons, which be transported so important that we gotta switch it to the next one. So many ways to pump protons. Shit, choose one. Now let it flow. Yeah. Now watch your ATP supply grow. Electron to the next one, on to the next, electron to the next one, on to the next, electron to the next one, on to the next, electron to the next one, on to the next. Full of freaks. Somebody synthesize some ATPs. Oxyphos is pretty complex. You can say how come wasn't so many steps in the mission outcome. Get that energy from the food that you eat was burning one big step, then you lose forest heat. So when those E minuses move down the chain, they make protons move across the membrane. And when the proflow gets to send, there's a rumbling to make ATP and be osmotic coupling. But as the electrons get past the long chain and help through the work, and lose a little oxy, so by the end, you gon' need an oxy to breathe, and keep it rolling like oxy. Friends making CO2, oxy-oxidative. Why you think we call cellular respiration? Burning out to make ATP stacks up. Oxygen present, electron to the next one. Now you oxidate to breathe. Somebody synthesize some ATPs. What's the point of all these? Trace tests, what a mess, make ATPs. Why you gotta do from glute to pyruvate, no ways to sedation? Why you think we need to breathe? Well, and then it's in carboxylate, baby. It ain't hard to tell, helps the cell do its food, eating chewy. You judge that it's crucial to me. Cause sodium to keep myosin moving, which is to say that it's fuel for your brain, your muscles, and they're just so insane. So when you think you're drunk, all take a test and drop these lows. Remember how you make ATP out of glucose. I call it Cisco chop. It goes down to 3C blocks. You can study me. I'll make ATP, and if you need it for reason, then you can get to know me. And you got enough oxygen, then stop. Mitochondria will do go ahead, study me. I'll make ATP. I'll make a hell of a lot, so you should get to know me.
Now you can say that you've taken a tour of Stanford's campus. Thanks. Good job, everyone. We're almost through with cellular respiration. We'll get to 9.5 and a little bit of 9.6 next.